Hello, good evening, everyone. So today we are in the week 4 tutorial session of the NPTEL course, Molecular Biology. So I will wait for 3 more minutes and I'll start at 7:05. Mm, Let's wait so that other learners can join. Okay, so I hope all of you have gone through the week 4 lectures. So in the week 4 lectures, we are now starting or the course in as we saw in week 4. Now we will there has been more focus on molecular biology. So we have what we saw in week 4 was what are the basics of we learned about the basics of molecular biology. So what is a genome, eukaryotic genome, prokaryotic genome, how the genome is packaged inside the nucleus, what are the what is the hierarchical model by which the DNA is folded inside the nucleus in eukaryotes. And then we also learned about the one of the main concepts of molecular biology, which is central dogma. Okay, central dogma of molecular biology. So now, gradually, in the upcoming lectures, we will deal about each of these processes in central dogma: replication, transcription, and translation. Okay. Uh, okay. So other than this, we also learned about giant chromosomes, the polyethylene chromosomes and the Lambrus chromosomes. Okay. So I'll start sharing the slides for today. Okay. So I hope the slides are visible. Okay. So yeah, welcome to the week 4 tutorial session of this NPTEL course, Molecular Biology. I am Akash Jyoti Pathak, and I'm one of the PMRFTS for this course, which is being taken by Professor Vishal Trivedi of IIT Guwahati. So yeah, I am a PhD scholar in the Department of Biosciences at IIT Bombay. I'm working in a structural biology lab. So let's first quickly recap what were the topics which were covered in week 4. So in week 4, first of all, we learned about the that because once it was once Mendel gave the concept of that there are some physical factors which are responsible for the heredity or which are responsible for inheritance of characters in from parents to progeny. The question that came forth was what are these factors? Are they DNA, or are they protein, or are they RNA? Okay, for answering these questions, several scientists performed several experiments. First was the Griffiths experiment, which was done on mice using this there are using the pneumonia strain, and then with this after following this, there was another group of scientists, Avery, McLeod, and McCarthy, who performed certain biochemical experiments to find out that what is the genetic material, and then the Hershey-Chase experiment, which the Hershey-Chase experiment gave the unequivocal proof that yes, DNA is the genetic material that gets transferred from parents to offspring, right? So all these experiments, if you had biology in class 11th and 12th, you must have learned about these experiments, and also in your BSc you learn about them. So we will discuss questions related to these concepts. Then we learned about the central dogma of molecular biology. How is the information or the genetic information flows in living organisms, right? From the DNA, the information which is present in DNA, it gets copied into mRNA, and then from mRNA, the genetic code which is present, it is translated into protein, right? Then we looked into prokaryotic and eukaryotic genome. How is the genome? What is a genome, and how is the genome of a prokaryote and eukaryote? What are the differences between them? How is the genome of a eukaryotic organism packaged inside the nucleus, right? And then we also learned about giant chromosomes. Okay, giant chromosomes. We saw about polytene. We learned about polytene chromosome and Lambrus chromosome. Okay, and a little bit about the replication also we had seen. So we had covered only the very basics of the, or we have just started with the molecular biology part. Okay. Okay, so now to answer this question first. So the catabolic pathways result in dash of the molecules. So this question, this was covered in metabolism. We had covered in week two. It's week two or week three. In week two, we had covered metabolism, right? Glycolysis, then Krebs cycle, fatty acid metabolism, all these we had seen, right? So this is a very basic question of metabolism. So here the question is, catabolic pathways result in dash of the molecules, and the options are assembly of the molecules, functionalization of the molecule, conformation, breakdown, right? So, so this in metabolism comprises of what they are: anabolism and catabolism, right? So anabolism and catabolism together is considered as metabolism. It's called metabolism. So in anabolism, what happens? In anabolism, in anabolism, synthesis of molecules takes place, right? Synthesis of various molecules or biomolecules like lipids, proteins, nucleic acid, synthesis of various biomolecules take place. And in catabolism, what happens? Breakdown of molecules, right? Breakdown of molecules take place in catabolism. So, so for synthesis, it is obvious that for synthesis, energy will be required, right? Okay. And in catabolism, what will happen? Energy will be released, right? In this case, energy is released, and the released energy is stored as ATP, NADH, FADH2. So they are they are stored as high energy molecules inside the cell. And then so that during anabolism, these high energy molecules can be used for synthesis of various molecules. Okay, so this is a very basic question. So metabolism, as you know, it is anabolism and catabolism, and in catabolism, breakdown of molecules into smaller products takes place, right? So the answer is D, option D. Okay, okay, so let's once revise what is metabolism. Well, so metabolism is the sum of all the chemical transformations taking place in a cell or organism. So all the chemical reactions which are taking place inside the cell, it can be both a formation of new molecules or breakdown of molecules. So together, this is called as metabolism. So all the reactions that we have seen, all the different steps of glycolysis, Krebs cycle, electron transport chain, and all these fatty acid metabolism, all these reactions will come under metabolism, right? So it occurs through a series of enzyme catalyzed reactions. So as we have seen when we were discussing glycolysis, there were several enzymes which are involved in each step of the glycolysis pathway of the Krebs cycle pathway. So all these breakdown and or synthesis, they takes place through a series of enzyme catalyzed reactions. And so what is catabolism? So catabolism is the degradative phase of metabolism. So as we discussed just now, it is the degradative phase of metabolism in which organic nutrient molecules, carbohydrates, fats, and proteins are converted into smaller, simpler end products such as lactic acid, carbon dioxide, and ammonia. Okay, so as we have seen in glycolysis, the glucose molecule is getting broken down into pyruvate. And then in the anaerobic reaction, this pyruvate gets further broken down into lactic acid. So that is an example of catabolism. Okay. Then when proteins will be broken down, ammonia, smaller molecules, ammonia, these will be released. So these are catabolism. So it is a degradative phase of metabolism. And so catabolic pathways, what will it do? It will release energy. So as we saw just now that in catabolism, because these molecules are broken down into simpler end products, there will be release of energy, some of which is conserved in the form of ATP and reduced electron carriers, NADH, NADPH, and FADH2. The rest is lost as heat. Okay. As we had seen, that energy is released in catabolism. Okay. Energy is released in catabolism, and this energy which is released, it is stored as ATP or electron carriers. So these electron carriers, they will be involved in the electron transport chain, and through the electron transport chain, when proton gradient is created, and when it is broken, again energy is released. So in anabolism, anabolism is opposite of, or it is exactly opposite of catabolism. It is biosynthesis, that is a degradation phase. This is a biosynthesis step. And here what happens, small simple precursors are built up into larger and more complex molecules, including lipids, polysaccharides, proteins, and nucleic acid. Okay. So different products which are produced, they are again involved in synthesizing large molecules like biomolecules, macromolecules, lipids, polysaccharides, proteins. So these will come under anabolism. Okay. So anabolic reactions will require an input of energy, right? So generally in the form of phosphoryl group transfer potential of ATP and the reducing power of NADH, NADPH, and FADH2. So this is a figure from the book Leninger. So here, as you can see, that it is shown through this figure that this anabolism and catabolism. So as you can see in anabolism, what is happening? In anabolism, there are these precursor molecules like amino acids, sugars, fatty acids, nitrogenous bases, and these precursor molecules are using this precursor molecule, larger macromolecules are generated utilizing. So here energy is utilized. ATP, the electron carriers which are there, they will be also utilized. And again, ATP will be produced. So utilizing the energy, higher macromolecules, proteins, polysaccharide, lipids, nucleic acids, these are produced. And, and in catabolism, these carbohydrates, fats, and proteins, which are present in our diet, for example, also these are again broken down. And when they are broken down, what we can see that here there is release of energy, and this release of energy is the released energy is stored as ATP or in electron high energy electron carriers, right? Reduced electron NADH, NADPH2, FADH2, and we get simpler products. Okay, simpler end products: carbon dioxide, water, and ammonia. Okay, so remember, catabolism is degradation or degradative pathway or degradation of uh, of large molecules into smaller molecules, and anabolism is biosynthesis. Anabolism basically means biosynthesis. Biosynthesis. So in degradation, energy will be released. Energy released, and here energy will be utilized. Okay, now answer this question. In some viruses, so we had learned about these concepts in the week 4. So try to answer this question. In some viruses, ribonucleic acid is present as genomic content. So you all know that viruses can have either RNA as the genetic material or DNA as genetic material, but not both. They either RNA is present as genomic material or DNA is present. RNA and DNA are not present together. So, in RNA viruses or viruses which are having RNA as the genetic material, in those viruses, what happens? This RNA is reverse transcribed using the enzyme reverse transcriptase. This RNA is converted into DNA. Okay, and so this process is called reverse transcription, and the enzyme used for this purpose. So, what is the enzyme? So you know there is an enzyme called reverse transcriptase. So there is another name for this enzyme. So what is the name of reverse
DNA polymerase.
RNA dependent DNA polymerase, and why? Why is it RNA dependent DNA polymerase?
So reverse transcriptase and RNA-dependent DNA polymerase, both mechanism are same.
Yeah, yeah, but why is it? Why is the name RNA dependent DNA polymerase? Because this reverse transcriptase enzyme, it will use RNA as the template, and it will, from using RNA as a template, it will generate the DNA, right? It will generate the
Yes.
DNA. So it is RNA dependent DNA polymerase. Okay. Similarly, DNA polymerase, you know the name of DNA polymerase? DNA
DNA
polymerase
dependent
are DNA dependent.
RNA polymerase.
RNA polymerase also has another name, and DNA polymerase has another name. So uh, can you tell me what is the other name of DNA polymerase and RNA polymerase? So basically, you just need to understand the concept. You do not need to memorize the names. Okay. So in DNA polymerase, what is happening? DNA polymerase is involved in replication. You know that, right? So in
Yeah.
replication, what is happening? DNA is getting replicated to form another copy of DNA, right? So the
Yeah.
enzyme, this DNA polymerase, will act on what? It will use DNA as a template, and it will generate another copy of DNA. So it is called DNA
DNA
dependent DNA
dependent DNA
polymerase.
polymerase.
DNA dependent DNA polymerase, because it is using DNA as a template and generating another copy of the DNA molecule. So it is DNA dependent DNA polymerase, and RNA polymerase is called. Now RNA polymerase will use. Ah,
Um,
right.
RNA RNA generating DNA. So
Ah,
RNA dependent DNA polymerase, like reverse transcription.
hmm. No, reverse transcriptase enzyme is called. So reverse transcriptase enzyme is called RNA dependent DNA
RNA
polymerase, right?
dependent DNA polymerase.
Ah. But RNA polymerase enzyme is called what? What is the other name of RNA polymerase? I am asking. This is RNA-dependent DNA polymerase. Yes, you are correct. The answer is option D. That is correct. RNA-dependent DNA polymerase. Okay. RNA polymerase, in RNA polymerase, what happens in transcription? One of the strands, the DNA strands, is used as a template, and using that strand as a template, the other strand is transcribed into its corresponding mRNA or complementary mRNA. So it is RNA dependent or it is DNA dependent RNA polymerase. RNA polymerase is also called DNA dependent RNA polymerase because it is using DNA as a template, and using DNA as a template, it is synthesizing its complementary RNA sequence, right? So RNA polymerase is called DNA dependent RNA polymerase because this, when you will learn the steps of transcription, you will see that this RNA polymerase binds to one of the strands of DNA, which is called the template strand. And using this template strand, the mRNA will be synthesized. Okay. So we'll learn about it. So you just have to understand the basic concept. So in DNA polymerase, the template is DNA. So that is why it is called DNA dependent DNA polymerase. In reverse transcriptase, the template is what? It is RNA. From RNA, the DNA is getting generated. That is why it is called RNA dependent DNA polymerase. And in RNA polymerase, DNA is the template, right? And using DNA, RNA is synthesized. So DNA dependent RNA polymerase. Okay. So these are the different names. So do not get confused in these questions. Okay. Okay. So yeah. So this was there in your slides, in your lecture slides. So I just I have copy pasted this slide from your lecture slides. So yes, reverse transcriptase is it is an exception or it is a special case of central dogma. So in reverse transcriptase, you know that the organisms which have RNA as a genetic material. So in these cases, what happens? RNA is converted into DNA. And how is it done? So basically, as we will see when we will discuss about central dogma, that genetic information flows from DNA to RNA and from RNA to protein. Okay, but in reverse transcription, so as the name reverse transcription means, it is reverse of transcription, means from RNA, you are generating DNA. So in this case, what happens? There is another enzyme which is called reverse transcriptase, and this reverse transcriptase converts the RNA into DNA. So that this DNA can then be integrated into the host replication machinery, and the RNA of the virus can be generated, and from mRNA, the virus can generate its own protein. Okay, so this is done by reverse transcription utilizing the sequence information of the parent RNA. Okay, so you have the parent RNA, and using the sequence of the RNA, the complementary DNA can be synthesized, right? The enzyme used for this purpose is RNA-dependent DNA polymerase. RNA-dependent DNA polymerase. Okay, now let's quickly review the Griffiths experiment. Okay, so would you like to explain what is this Griffiths experiment? You have already learned about the Griffiths experiment in class 12. So would anyone like to explain the Griffiths experiment? Okay, so Griffith performed a simple this microbiology or biochemical experiment to find that what is the genetic material that gets transferred uh, from parents to offspring. Okay, so what is the model system that he used? So he used salmonella bacteria as his model system. Okay, and if you see salmonella pneumonia, so this salmonella bacteria has two different strains. One is the S3 strain and another is the R2 strain. So there are two strains of, there are other strains also, but he focused on these two strains, R2 strain and S3 strain. And this S3 strain, so if you look at the morphology of the colonies, which the S3 strain have, they have smooth colonies. They have smooth colonies, and the R2 strain have rough colonies. if you plate them on a agar plate, you will find that these colonies, R2 strain Salmonella, have rough colonies, and S3 colonies, they give a smooth appearance. Okay, and why do they have smooth appearance? Because they have a covering over the cell wall, or which is the glycocalyx covering over their cell wall, and which is why they give they appear as smooth colonies. And because of this glycocalyx covering, they are virulent. They are virulent means they have the potential to cause infection in a living organism. While this R2 strain, they are avirulent. Avirulent, which means that they cannot cause disease. Okay. Virulent. Okay. So now what he did is that so Griffith observed that small amounts of living type S3 bacteria injected into mice caused the mice to develop pneumonia and die. So that is what I was explaining. So this S3 strain of Salmonella, they are virulent. So when this S3 bacteria is injected into the blood of a healthy mice, the mice develops the infection, pneumonia infection, and it will die. So this is the first observation or first experiment that he where he found that when this S3 strain of bacteria were injected into the mice, the mice died. Okay, and what he found when he isolated when he did an autopsy and isolated the blood from this dead mouse, he found that this blood contained virulent bacteria. Now, this is the R2, or the non-virulent strain of the bacteria. So when this R2 strain of bacteria was infected into the healthy mice, it was found that the mice did not die, it was alive, and also no bacteria was recovered from the blood of the healthy mice, or the mice which did not die. Now, what he did in the next step, he he heat killed the S3 strain of bacteria. Once you heat the S3 or once you heat the bacteria will die. It will it will lose its property to cause infection, and so he makes the heat killed S3 bacteria with the live R2 bacteria, and now he injected this into the healthy mice. And what he found that because these were dead, because the S3 strain was heat killed and the R2 did not cause any infection, he found that the mice was alive, and no bacteria was recovered from the blood of this alive mice. Now, what he did that when he now what he did, a mixture of type R3, R2 bacteria and heat killed type S3 bacteria are injected into a mice. No, sorry. So in this C exam, only heat killed S3 bacteria was injected into the mice. So only heat killed S3 bacteria is injected into mice, then the mice will stay alive. Now, but if when he makes this S3 heat killed S3 bacteria with the live R2 bacteria, he found that the mice developed the infection, and he and died. So this was quite a striking observation. Why was this striking? Because the S3 strain of bacteria was heat killed, and the R2 bacteria did not have the ability to infect or cause disease. But when these two strains of bacteria was mixed, it was found that the mice died. So now, and when an autopsy or the blood of this dead mice was taken, it was found that he would find what he found, that there were he isolated S3 bacteria from the blood of the dead mice. So so this is so. So what? How did he explain this observation? So when Griffith injected type R2 bacteria into the mice lived and no bacteria were recovered from the blood. When he injected large amount of heat kill type S3 bacteria into the mice lived and no type S3 bacteria were recovered from their blood. Okay, so this was used as a control, right? So because when this S3 strain of bacteria, heat killed S3 strain of bacteria was mixed with the R2 strain of live bacteria, the mice died. So could there be any error in this heat killed S3 strain of bacteria in the injection, or whether any during the experiment any error has been created. So this was used as a one of the controls. But when he found that when S3 heat killed S3 strain of bacteria was used, the mice was alive. So Griffith unexpectedly found that the mice injected with live R2 bacteria and heat killed S3 bacteria developed pneumonia and died, with live heritable S3 bacteria recovered from their blood. The S3 bacteria was heat killed, but when the blood from the dead mice was taken, it was found that the S3 bacteria he could isolate live S3 bacteria from the blood. So, how did he conclude? He concluded that a substance in the heat killed virulent bacteria genetically transformed the type R2 bacteria into the live virulent type S3 bacteria. So, you must have read about transformation, or you must have done in your practicals also this transformation experiment that the bacteria has a property. A bacteria has a property to take up DNA from the surrounding environment. So when this S3, what happened is when this S3 strain of bacteria was heat killed S3 strain of bacteria was mixed or was mixed with the R2 strain of bacteria, the DNA from the S3 strain of bacteria was taken up by the R2 strain of bacteria, and then this R2 strain of bacteria because it got converted into the S3 strain of bacteria. So they could take up the DNA. They could take up the DNA from the S3 strain of bacteria, and then this R2 strain of bacteria develop the S3 type of colonies. Okay. So transformation is the method is one of the method by which bacteria can reproduce. There are other methods, conjugation, transduction. So transformation is one of the method. So this is the method by which even if you are working in a lab, you must have this is a very routine experiment that is done in lab where the plasmid is plasmid uh, is transformed into a competent cell. So you create competent cells of different bacterial strain, and then once you add the plasmid, there are some other steps, um, heat shock, and by through these steps, the bacteria can easily accept the DNA from the environment. Okay. So what is the final outcome of the Griffith experiment? So one by one, we will see the option. So R2 heat killed mice dead. So if you inject heat killed R2 strain into the mice, then the mice will it be dead or will it be alive? So this option is wrong, right? The mice be will remain alive. Okay. This option is wrong. If you inject S3 live bacteria and R2, so mix S3 live bacteria and mix it with the R2 heat kill bacteria, then the mice will stay alive. Is this option true? This option is also false because you are injecting because it has S3 strain of bacteria. Now S3 strain of bacteria is virulent. So if S3 strain of bacteria is injected into the mice, then the mice will be dead. Then option C, you have mixed the S3 heat kill bacteria with the R2 live bacteria, and then you have injected it into the mice. So you find that the mice is dead. So this was the striking observation that in Griffith's experiment, right? So the expected outcome would be that the mice will stay alive because this S3 strain is heat killed and the R2 strain is alive, but the R2 strain is virulent. So the mice should have stayed alive. But the main observation or the striking observation was that the mice was dead and he could recover living S3 strain from the blood of the dead mice. So this is option number C is correct, and option D is what? S3 heat kill strain mixed with R2 live strain, and if the mice was dead. So heat killed S3 and R2 live. So this is the same option actually, option C and D. There is an error in this question. Option C is correct basically. Now coming to the genome. So what do you understand by genome of an organism? So the total haploid content of so the total haploid content of an organism or haploid content of DNA is called the genome. Okay. So for human, the genome is what? The genome is 6 billion base pairs. Okay. For bacteria, it is around 4.6 million base pair. It's not. It is 6 billion is not the haploid content. 3 billion. Okay. So the complete set of DNA comprising the nuclear and mitochondrial DNA is called the genome of an organism. So prokaryotes have small circular DNA present in the nucleoid region. So this is the this is the haploid content, but in eukaryotes, the chromosomes are present in sets of two, right? So that will be so the total it will be 6.6 billion base pairs, right? 6.6 billion base pair, but this is not the this is the diploid content. But genome by genome we mean the haploid content of DNA. So it is 3 billion base pair. So prokaryotes have small circular DNA present in the nuclear region, and they have single chromosome. Prokaryotes have single chromosome. They have most of the bacteria. They have covalently closed circular DNA, as we were discussing in the last lecture also. And the eukaryotic genome is so the eukaryotic genome is monocystronic, while prokaryotic gene is polycystronic. So you will learn about these terms in your when we in the later lectures when transcription and lac operon concept, all these will be discussed. You will get know about, you will understand these terms monocystronic and polycystronic. So for now, just remember prokaryotic means polycystronic and eukaryotic genome are monocystronic. So basically, when you will learn about the mRNA, so mRNA has a promoter region and a terminator region, right? So if the gene which is present is has only one promoter or it is present between a promoter and an operator, then it is called monocystronic. While if there are multiple genes which are present between a single promoter and operator, promoter and a terminator,
আমি
promoter
চলে
and
গেছি না স্যার, তবু একা একাই বকে গেছে, কি অদ্ভুত।
so between a promoter and terminator sequence, if there are multiple genes present, then then in that case it is called as polycystronic. So polycystronic gene is present in prokaryotes, while eukaryotes have eukaryotic genome is monocystronic. Okay. So 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 during when you will learn about transcription, you will learn about these concepts that there are specific sequences on the DNA where the RNA polymerase binds. These sequences are known as promoter, and there are specific sequences on the DNA where where the DNA polymerase falls off from the coding region of the DNA. So those regions are called the terminator. So if a gene is present, if only a single gene is present between the promoter and terminator sequences, then it is called as monocistronic. So monocistronic is present in prokaryotes. Monocistronic is present in eukaryotes because there is only a single gene is present between a promoter sequence and a terminator sequence. Okay. So the gene, if you see the coding region of a DNA, in a coding region of a DNA, only a single gene, if only a single gene is present between a promoter and a terminator region, then this type of genes are called monocistronic gene. And if there are multiple genes, so there is a promoter sequence. So we'll learn about this promoter sequence, terminator sequence. So between the promoter sequence and terminator sequence, if there are multiple genes present, gene 1, gene 2, gene 3. So in those cases, this is called polycistronic genome. Okay. Okay. So the eukaryotic genome is linear and the prokaryotic genome is circular. We are seeing this already. And another thing is that in eukaryotes have exon intron organization. So if you so you will learn about this concept. There is exon intron. So the mRNA has alternating regions of exon intron. So these introns are chopped off from the mRNA, and then exons are. So this is splicing step. So all these steps you will learn. So just for now, you remember that these genomes are eukaryotic genome have alternating exon intron regions, while the prokaryotic genome do not have introns between this or intron regions are not present here. Okay. Okay, now answer this question. Which one is the characteristic feature of the eukaryotic genome? Contain a single set of chromosome. Am I audible? So which one is the characteristic feature of the eukaryotic genome? Eukaryotic genome contains a single set of chromosome. Is this correct? You know, we all know that eukaryotes are diploid organisms, right? So eukaryotes most eukaryotes have two sets of chromosome. We have in humans there are 46 chromosome or 23 pairs of chromosome. One chromosome is the paternal chromosome and another chromosome is the maternal chromosome. So there are always two sets for each chromosome. mRNA has a short lifespan of a few minutes. For eukaryotic genome, is it true? This is not true actually. Okay. Polycistronic. Just now we learned about the concept of polycistronic and monocistronic. Polycistronic is genome is present in prokaryotes. So remember P and prokaryotes have P. So polycistronic means prokaryotes because there are multiple genes which are controlled by a single promoter region. And the prokaryotic genes are polycistronic. So this is also incorrect. Whole genome does not code for a protein. So as we know that if you look at the entire DNA, only a small region of the DNA, there are specific regions in the DNA which codes for a protein or specific regions in the DNA which gets transcribed into our mRNA, and this mRNA is then translated into protein. The whole genome is not transcribed. If you this entire is this 3 billion base pairs are not transcribed. There are specific regions of the genome which are transcribed. Okay, so yes, answer is option D. So this is directly from one of the slides, from one of your lecture slides. So prokaryotic genome, they have small genome, no specialization. Eukaryotic genome are larger, and they are specialized. They are more with they have uh, specialization. Prokaryotic genome have DNA plus few proteins in simple arrangement, while the eukaryotic genome, the DNA is wrapped around a complex of protein, as we will see in the later slides. There is only a single set of chromosome for prokaryotes, and while eukaryotes have one or more sets of chromosomes. The amount of DNA is smaller, they're typically greater than that in bacterial cell. They are polycistronic, they are monocistronic. Most of the DNA codes for proteins or RNAs, very little junk DNA. So the part of the genome which does not code for any DNA, does not code for any protein is called as the junk DNA. So this junk DNA is very profoundly found in eukaryotic genome. So most of the DNA does not code for protein or RNAs. RNA processing not an option for controlling expression. RNA processing allows for several opportunities to regulate genes. So we will learn about RNA processing, the splicing, and what are the different proteins involved in splicing. So we will learn about it. And mRNA has a short lifespan in prokaryotes, while mRNA is long-lived in eukaryotic genome. Okay. Hello. Okay, we will move ahead. The nucleic acid type in tobacco mosaic viruses. So tobacco mosaic virus is a plant virus. It is a plant virus, which means that it infects plants. So tobacco mosaic virus is a plant virus. So as we had seen that, what is it is a plant virus, and as we had seen that viruses have can have either RNA as the genetic material or DNA as the genetic material. There are some viruses which are which have single-stranded RNA. There are viruses which have single-stranded DNA, double-stranded RNA. So you have to remember the name of these viruses. So tobacco mosaic virus is a very common virus, and there are several experiments which have been done using tobacco mosaic virus. So usually plant viruses have single-stranded RNA. So the correct answer is single-stranded RNA. So you remember that tobacco mosaic viruses have single-stranded RNA. Okay. So this is again from one of the lecture slides. The virus consists of two parts: nucleic acid and protein code. As we have, if you look at the structure of viruses, then the genetic material is present in the central code, and this genetic material is surrounded, or there is a protein code which is known as capsid, which is present surrounding this genetic material. And these viruses contain only one type of nucleic acid, either DNA or RNA. So only they can have DNA or RNA. Both are not present in viruses, and viruses having RNA, they are called as riboviruses. They vary in structure of their nucleic acid. Most of the plant viruses have RNA, either single-stranded or double-stranded. So you remember this point that plant viruses mostly have RNA as the genetic material. Okay, so here there is an example. So animal RNA virus, the having single-stranded RNA. Animal virus is picorna virus, toga virus, and double-stranded RNA virus. Animal virus is Rio virus. Okay, while in case of plants, the single-stranded plant RNA virus is tobacco mosaic virus, while double-stranded RNA virus is Oryza virus. So just remember these few examples. That will be good enough. Now, coming to the genome organization in eukaryotes. Now, if we look at the genome of the genome organization in eukaryotes, So the DNA or the genome of a human genome is very long, right? It is the human genome is has around 3 billion base pairs. Okay, or the total, if you consider the double deployed content is 6 billion base pairs. So it is 6 into 10 to the power 9 base pair. And if you calculate the length of the DNA, if it is completely stretched out, considering that, yeah, each base pair, what is the distance between each base pair? Distance between each base pair, as we have seen, it is around 3.4 angstrom, right? Okay, so if you do this calculation, then if you will find that if the eukaryotic DNA is completely stretched out, it will extend to around 2 meters. It will extend to around 2 meters, while the nucleus in which this DNA is present is only a few micrometers in diameter. So it is around 10 micrometer in diameter. So if this 2 meter long macromolecular structure has to be organized, or it has to be present inside this 10 micrometer diameter of the nucleus, there must be some packaging must be done, which will allow this such a long structure to be accommodated inside the nucleus. And this packaging is done by several proteins around which the DNA wraps around. So the eukaryotic DNA in the cell is closely associated with proteins, and these proteins help in packaging the DNA into packaging of the DNA, and the structure which is formed by the combination of this DNA and protein is called chromatin. It's called chromatin. Okay, so the most abundant protein in chromatin are the histones, which are small positively charged proteins of five major types. So there are five major. So the proteins which are involved in packaging of the DNA are called the histone proteins. And if we look at these histone proteins, there are five major types of histone proteins, which are H1, H2A, H2B, H3, and H4. Right. So out of these H2, H2B, H3, and H4, these are called the core histones, because these histones are present in copies of, there are two copies of each of this H2, H2B, H3, H4, and together they form a complex which is known as the histone octamer complex. And around this histone octamer, the DNA is wrapped around about around 147 base pair of the DNA is wrapped around this octamer complex, and the and forms a structure which is known as the nucleosome. And and this H1 is not a it is not a core histone, but it is also known as linker histone. And this histone protein is involved in compaction of the DNA, or it is also uh, involved in involved in sealing or of the entry and exit sites of the DNA. Okay. So all histones have a high percentage of arginine and lysine amino acid residues. Why? As we have seen that arginine and lysine are basic amino acid residues, and they usually have a positive charge at the physiological pH, because, and since they have a positive charge and DNA have negative charge because of the phosphate groups, these amino acid residue or basic amino acid residue will allow the DNA to wrap around these histone octamer complexes. The positive charge attract the net charges on the phosphates of DNA. The chromatin isolated from the nucleus of a cell looks like beads on a string when viewed with an electron microscope. So if you isolate the DNA or the chromatin from the nucleus of a cell and look under the microscope, you will see that this chromatin have a will give a bead on a string structure in the electron microscope. So the beads are called the nucleosome. These are called the nucleosome, and these are the linker DNA. So these regions, those are called the linker DNA. So the nucleosome is a core particle which consists of DNA wrapped about two times around an octamer of 8 histone proteins. So there are two copies each of these H2, H2B, H3, H4. So total there are eight number of these histone proteins. There are 8 histone proteins. Nucleosome folds on themselves to form a dense, tightly packed structure that make up a fiber with a diameter of about 30 nanometer. So these nucleosomes which are formed, this is the first order folding of the DNA. The primary structure of the structure of the folded DNA. This is called the chromatin, and these beads are called the nucleosome, around which this DNA are wrapped. These are called the nucleosome. Now, these nucleosomes form a connection with the adjacent nucleosome or internucleosome. There is interaction between the internucleosomal structures, and they associate with each other to form a higher order structure, which is called as the chromatin fiber. This chromatin fiber has a diameter of about 30 nanometer, and there are different models which have been proposed for how the DNA is folded in a chromatin fiber. So one is the zigzag model, and another is the solenoid model. So in solenoid model, a linear array of nucleosomes are coiled, and a helix model in which the nucleosomes are arranged in a zigzag ribbon that twists or supercoil. So this is the figure, one of the figure which I've taken from the genetics book, Pierce. Here, what you can see here, that this is the primary, or that is the simplest level. At the simplest level, the chromatin is a double-stranded helical structure of DNA. So as we have seen that the DNA has a diameter of 2 nanometer, right? So this is the simplest level of folding. Now, this DNA gets wrapped around by histone octamer. Is wrapped around by wraps around histone octamer and form a structure called nucleosome. So the DNA is complexed with histones to form nucleosomes, and each nucleosome. So if you look at the structure of nucleosome, then this nucleosome consists of 8 histone proteins. So there are two copies of H2, H2B, H3, H4, and the DNA wraps around about 1 whole 3/4 turns, or 1.65 times. The DNA is wrapped around the histone octamer, and this nucleosome associates with H1 protein. So this H1 protein and nucleosome, this together, they are called the chromatosome. So the chromatosom consists of a nucleosome plus the H1 histone, and the next level of folding is this. You can see in this figure, these nucleosomes associate with other nucleosomal structures, or they form very close connections with their neighboring nucleosomes and produce a 30 nanometer fiber. Right. And then this 30 nanometer fiber, there are, other than histone proteins, there are several non-histone proteins or histone chaperones. These proteins come together, and they help in forming of higher order structure of the chromatin. So that forms the 300 nanometer structure, and then this 300 nanometer, the 700 nanometer structure is produced, and further folding or compaction gives rise to ultimately the 1400 nanometer structure, which is known as the chromosome. So if you look at the chromosomal structure, it has two chromatids which is joined at the center. This is known as the centromere, right? So the tight coiling of the 250 nanometer fiber produces the chromatid of a chromosome. Okay, now answer these questions. Which one is the correct match of the following? So you have uh, uh, this on one side, you have these four options in capital letters A, B, C, and D. You have to match it with this small letter A, B, C, D options. Okay. So nucleosome code, option A, not option A. This is the first column, nucleosome code, bead on a string. Then C is 700 nanometer chromatin fiber. And D is what? It is 30 nanometer chromatin fiber. And on the other column, you have four choices, right? Nucleosome, higher order packed chromatin, then there is this solenoid chromatin fiber, and option D, and the choice number D is 145 base pair DNA. So you have to match column A with column B, or the statements in column A with these options in column B. So in the nucleosome code, we just now saw that the nucleosome code is found by 145 base pair of DNA, right? 145 base pair of DNA wraps around the histone octamer. Beads on a string is what? So the beads are the nucleosome basically, right? These are the beads on the string are called the nucleosome. The 700 nanometer chromatin fiber, it is the higher order packed chromatin, right? Higher order packed chromatin, 70 nanometer, 700 nanometer chromatin fiber, and the 30 nanometer chromatin fiber. There are several models given for this 30 nanometer chromatin fiber, which are the solenoid model and the helix model. So of this D will match with C. So now you can answer this question. A is matching with D, B matches with A, C matches with B, and D matches with C. Isn't it? A, D, B, A, C, B, D, C. So the option number B is correct. Right? Option B is correct. So this is the hierarchical folding of DNA into higher order chromatin structures. So this is one of the model by which we can explain that how this 2 meter long structure remains packed inside the 10 micrometer diameter of the nucleus. Okay. So what are the thread-like stain structure present in the nucleus known as? So we had already seen this in week one tutorial session. So what is a chromatin? What is a chromatid chromosome? So all this we had discussed briefly in week one. So the thread-like stain structures present in the nucleus are called. So if you look at the interphase nucleus, then the DNA is present as a thread-like structure, right? So this thread-like structure is called the or this array of nucleosome which is present in the DNA, or that when this DNA assumes a structure which is which assumes a form of array on a array of nucleosome. This is called the chromatin. Okay, so chromatin is the answer. So chromatin means basically DNA plus histone proteins together. The DNA plus nucleosome is called the chromatin. So array of nucleosomes is called the chromatin. The structure which is formed by arrangement of array of nucleosomes side by side is called the chromatin. Okay. So how many types of histone proteins are present in the chromosome? So types of histone proteins. So there are 5 types of histone proteins. So you have to read the question carefully whether it is what are the types of histone proteins or what are the types of core histone proteins. So if it is core histone proteins, then there are only 4 types of histone proteins, which are H2, H2B, H3, H4. While if the question is how many types of histone proteins are present, then in that case it will be 5 types of histone proteins, right? H1, H2A, H2B, H3, H4. So there are 5 types of histone proteins. So the answer is option B. Okay. Is my screen visible? I hope these concepts are clear. These are very fundamental questions. Okay. So now we will learn about the central dogma of molecular biology. So what do you mean by the central dogma of molecular biology? It basically means how the genetic information flows inside the cell of a living organism. So the genetic information means that whatever information that is present in the DNA, how does this information get encoded and give rise to a particular phenotype that we see in an organism. So how does this information from DNA code for a protein. That also you can say that how the information is DNA is encoded and translated into to form macromolecules called protein. Okay, so this is so central dogma is basically the flow of genetic information in the cell, and this term central dogma was given by Francis Crick. So if you remember that Watson and Crick, they had solved the or they had given the double helix DNA model, proposed how does the a bDNA or the double-stranded DNA structure look like. So based on the structure that they had proposed, so from that structure, they had said that they had given this concept of central dogma. So what does this central dogma state? It states that the genetic information passes from DNA to protein in a one-way information pathway. So this is the DNA, and in the DNA, the sequence information gets this DNA is transcribed into RNA, and then this the information which is present in RNA that is translated to form structures called protein. Okay, so information is transferred. So it is a one-way information pathway. Okay, so information is transferred from DNA to an RNA molecule. Transcription. This is called known as transcription. And what does translation mean? Translation means that information is transferred from RNA to a protein through a code that specifies the amino acid sequence. Okay. So the transfer of information from RNA to protein through a code which specifies the amino acid sequence is called as translation. Okay. And what is DNA replication? From one DNA molecule is copied into another to form another DNA molecule is known as DNA replication. Okay, so a replication is the information passes from one DNA molecule to the other DNA molecules. Transcription is information passes from DNA to RNA, and translation information passes from RNA to protein. So this is the central dogma of molecular biology, which means that the genetic information will flow from DNA to RNA to protein. So it is a one-way information pathway. So there are some special cases of central dogma. So one is this reverse transcription, which we just learned in the previous slides. Not only the DNA can get converted into RNA, but there are certain organisms where we can see that the RNA can be used as a template for synthesis of DNA molecules, as was seen in retroviruses or RNA viruses. There is an enzyme called reverse transcription and through the process of reverse transcription, the RNA can get converted into DNA. So in some viruses, the information is transferred from RNA to DNA, or so these figures are mostly from the book genetic books peers. You can refer to the book. So uh, this is one of the standard books which are followed in um, many universities and colleges. This is a reverse transcription, and if the information from RNA copied into another RNA molecule, this is known as RNA replication, right? RNA replication also will also take place in viruses, right? Because viruses have RNA as a genetic material, and from one RNA, so RNA replication is not usually found in eukaryotes. Okay, in eukaryotes, prokaryotes, that they're mainly found in viruses, because these are they have the RNA as the genetic material, and this RNA can be copied from another RNA molecule. This is called RNA replication. So reverse transcription is found in retroviruses, and some transposable elements transfer information from RNA to DNA. And in RNA viruses, information can flow from transfer from RNA to RNA, which is known as RNA replication. Okay, now answer this question. So this is an interesting question. So you have to just name the process by which. Look at this question carefully. So RNA is getting converted into DNA. This process named as X. Now DNA is converted into DNA. This process is named as Y. DNA converting into mRNA named as Z. mRNA to protein is named as T. So you have to name the process by which RNA is gets converted into DNA. So the process by which RNA gets converted to DNA is just now we saw that RNA to DNA conversion is known as reverse transcription. It is known as reverse transcription. Okay. This is known as reverse transcription. DNA to DNA. DNA to DNA is known as what? This process is known as replication. This is known as replication, DNA to DNA. DNA to mRNA is known as transfer of information from DNA to mRNA is known as what? It is known as transcription. And mRNA to protein is known as translation. So your X is reverse transcription, Y is replication, Z is transcription, and T is translation. So option B is correct. So you have to understand the central dogma of molecular biology, how the genetic information flows from DNA to proteins. So we will look at all these different these different processes in greater detail as we will move ahead in the course. Okay. Now, so this I explained just now. In some viruses, ribonucleic acid is present as genomic content, and it can be converted into deoxyribonucleic acid. This is done by reverse transcription, the enzyme used for this purpose. So we just now looked into this question in the earlier slide. This is RNA-dependent DNA polymerase. Okay. So yes, so this is the figure of a retrovirus or a RNA containing virus. So you can see that RNA viruses, if you see their genome, they have single-stranded RNA, and they can also have double-stranded RNA. This is an example where of a virus which has single-stranded RNA as its genome, as a genome, and this single-stranded RNA also is associated with the reverse transcriptase enzyme. So due to the presence of this reverse transcriptase enzyme, once this virus infects the host using the host cellular machinery, the RNA can be reverse transcribed to produce DNA using this reverse transcriptase enzyme. Okay, so because the retroviral genome is RNA, okay, whereas that of host is DNA, a retrovirus must produce reverse transcriptase, an enzyme that synthesizes complementary DNA from either an RNA or a DNA template. Okay, so the host genome is of DNA, and so if this basically what viruses do is they integrate their genome into the host genome. So for integrating their own genome into the host genome, this RNA has to be reverse transcribed to form DNA. So once that is reverse transcribed into its complementary DNA, now this DNA can get integrated into the host genome so that when the host cellular machinery is being used for replication and transcription process, along with the synthesis of the host proteins, its own protein or the viral proteins will also get translated. Okay, so this is one of the mechanism by which viruses use for reproduction inside the host cell. So a retrovirus uses reverse transcriptase to copy its RNA genome into a single-stranded DNA molecule, which it then copies to make double-stranded DNA. So first from this RNA molecule, a single-stranded DNA molecule is made, and from this single-stranded molecule, double-stranded DNA can be easily made using the DNA polymerase enzyme. Here, reverse transcriptase enzyme will be used, and now this double-stranded DNA gets integrated into the host genome. Now, coming to one of the very important experiments, which was done on replica, which was done for identifying what type of replication is present in eukaryotes. Okay, so whether it is this is known as the Michelson style experiment. Michelson. experiment. So once it was confirmed that DNA is the genetic material, so by that time the structure of DNA was also solved, and it was known that the DNA is a double helix structure where there are two strands of DNA, two polynucleotide strands wrap around each other. There are two polynucleotide strands which wrap around each other, and the DNA is the genetic material which is present in most organisms. So now the question arises that how from one copy of DNA, two copies of DNA can be produced, or how does the replication of DNA takes place in living organism. There were several models which were put forth to understand for explaining how DNA replication takes place. One of the model was the so there were three main models which were proposed. First is the conserved. One is the conserved mode of replication. Second is the semi-conservative mode of replication, or conservative mode of replication, semi-conservative mode of replication, and the third is the disruptive or dispersive mode of replication. Okay. So what is meant by the conservative mode of replication? So in conservative mode of replication, the hypothesis was that this DNA molecule, this DNA molecules which are made up of two strands. Okay. So this DNA molecule acts as the template. Both these DNA strands are acting as a template, and from here, one there is one parental strand and one more DNA molecules which have both the parental strands, and another DNA molecule will be produced which will have completely two new strands. So these are two new strands. So the conservative mode of replication means from one DNA molecule you will get two DNA molecules. One DNA molecule will have both the strands as parental strands, and the other DNA molecule, in the other DNA molecule, both the strands will be newly produced. Both the strands will be newly produced. Now, so this is the conservative mode of replication. In semi-conservative mode of replication, it means that during replication, the two strands of DNA, the two polynucleotide strands, they will separate. Okay, so semi-conservative basically means that two strands will separate, and using these separated strands. So each strand will now act as a template, and a new strand will be synthesized. Okay. A new strand will be synthesized. So each DNA molecule that will be produced will have a parental strand and a daughter strand, or a newly synthesized strand. Newly synthesized strand is called as the daughter strand. Okay, and in dispersive mode of replication, the idea was that, or the hypothesis was that this DNA molecule. This is the parental DNA molecule. Okay, this parental DNA molecules, it is broken into very small pieces. It gets broken down into very small pieces, and then these fragments gets joined by a formation of new strands. Okay, so new strands are produced by yeah, new strands are pres or the old strands are interspersed by new daughter strands. So that was the idea for dispersive mode of replication. Now, to understand that which is the correct mode of replication, Missels and Stell performed an experiment they had used E. coli bacteria as a model system, and what they did here is that they have used they have what they have done is they have cultured E. coli cells. Okay, they have taken E. coli cells, and then these E. coli cells have been cultured in a media which is having N15 as the nutrient medium. Okay, so the salts, the glucose, whatever components are present in the media, they are all N15 labeled. Okay, they are all N15 labeled. If you culture the E. coli in a media which has N15 as the glucose source and the carbon source for several generations, then the E. coli which will be produced, they will have the DNA the proteins and whatever biomolecules which gets generated in the E. coli, they will have N15, they will be N15 labeled. Okay, so basically their DNA will be N15 labeled. Means both the strands of the DNA will have N15, will be N15 labeled. Okay. So DNA, as you know, they are made up of nitrogenous bases, so they will be N15 labeled. Now, these E. coli, which have been cultured for several generations in N15 media, they are transferred into a media having N14 as the nitrogen source. Okay, N14 as a nitrogen source. So you can see here in this figure that this the bacteria cultured in N15 media, they were transferred to N14 media and allowed to replicate. Now, there after replication in they have been cultured for several generations. So in each generation, they have been cultured in N14 media, and the DNA which was produced, the DNA have been analyzed by using equilibrium gradient density, density gradient. Yeah, equilibrium gradient density centrifugation. So if you isolate the DNA from the bacteria which have been cultured in N15 media, what was found that this DNA, when you spin it in a cesium chloride density gradient do a cesium chloride density gradient centrifugation, what was found that these bacteria, the DNA produced a single band, right? It produced a single band, which was N15, corresponding to the N15 region or the N15 label. Now, when this bacteria were transferred into N14 media, the DNA, the band was found to come at a region which is intermediate of the N14 and the N15 regions. Now, in the third generation of bacteria, when they were again cultured in this N14 media, two bands were found to be were observed. One having the N15, which is at the intermediate position between the N14 and N15, and another which is at the N14 position. Now, further culturing it for several generations, it was found that the intensity of the band, which is present at the N14 region, was increasing, while this band, which is at an intermediate position between the N14 and N15 region, the intensity was decreasing. So that is what is explained in this figure. So the DNA from bacteria that had been grown on media containing N15 appeared as a single band. So it is a single band. So after one round of replication, it was found that after one round of replication, a single band was observed. A single band was observed, and this single band was at a position which is intermediate between the N14 and N15 band at an intermediate weight. Now, after a second round of replication, DNA appeared as two bands, one light and the other intermediate in weight. So one is light, which is produced at the which so with the one which is light will remain at the top of the top of the tube, which is heavy, which will settle down at the bottom, right? So a band was produced at the one light band was produced and another intermediate band was produced. And then after another round of replication, after two rounds of replication, uh, again there were two bands were observed, one at an intermediate position and one at the light position. Okay. So from this experiment, it was confirmed that the mode of replication in eukaryotes is semi-conservative mode. Okay, so if it had been conservative mode, then can you tell me what will be the band that will be observed if it is conservative mode of replication? So in conservative mode of replication, you will obtain two bands. One is this N15 band, another is N14 band. You will not obtain a band at an intermediate position. So for conservative mode of replication, one band will be obtained at the bottom and another band will be obtained at the top. Because in conservative mode of replication, just now I explained that both of these strands will remain as it is, and from this another new strand of the two new strands will be produced. So there will be N15 will remain as such, and another N14 will be produced. So there you won't observe any intermediate or any bands at the intermediate position between the N14 and N15. Okay. Now if it was dispersive, If it was dispersive, what type of band would you observe? In case of dispersive, you will only observe an intermediate band. This is the band that you will observe if it was dispersive mode of replication. You will not observe any band at the light position or at the heavy position. It will be always remain at the intermediate position. But if it is semi-conservative, only in case of semi-conservative you can explain this observation that you are getting a band which is increasing in intensity at the light and 14 position, while this intensity of band at the intermediate position is getting decreased. So with further rounds of replication, you can see that the DNA molecules which are getting produced, they have both N14 N14 DNA strands, or both the strands are N14 labeled. And and the number of bands which are, or number of DNA molecules which are having N15 and N14 DNA strands are getting lesser and lesser. So Messelson, through this experiment, Messelson and Stell convincingly demonstrated that the replication in E. coli is semi-conservative. What is meant by semi-conservative? It means that what happens during replication, the two strands of the DNA separates, and each strand will act as a template for the synthesis of a new DNA strand, which is known as the daughter strand. So that is semi-conservative mode of replication. Now, after all this explanation was for you to answer this question, which is what is the mode of replication in E. coli? So the mode of replication in E. coli, in humans, in most organisms, the mode of replication is, in all organisms, it is semi-conservative mode of replication. So there are several options here given: intermediate, dispersive, conservative, semi-conservative. So for explaining this question, I have given this figure. This is from again from the textbook, this textbook by Pierce. And here it has been explained that what would be the observation if the mode of replication was conservative or dispersive. So as you can see that if it was conservative, so in conservative replication, the entire double-stranded DNA molecule serves as a template for a whole new molecule of DNA, and the original DNA molecule is fully conserved during replication. So this original DNA molecule is fully conserved during replication. So you can see, so this original DNA molecule, this is remaining as N15 N15, okay? And here N14 N14, you are getting other N14 N14 bands. So you will see that the lighter bands, the intensity of the lighter bands will increase, while the heavier bands, the intensity will get decreased. So this would be the observation for conservative replication. If it was conservative replication. In dispersive replication, both nucleotide strands break down. So these nucleotide strands, they break down or they get fragmented, and then these fragments act as template for the synthesis of new DNA fragments, and then somehow reassemble into complete DNA molecules. So somehow that is not known, but somehow this could be one of the method by which replication is taking place. It is breaking down into fragments, and then they are acting as templates for the synthesis of new strands on new nucleotide fragments. So resulting DNA molecules is interspersed with fragments of. So the resulting, you see here, the the DNA molecules that are getting produced, new DNA molecules that are produced, they are interspersed with fragments of old and new DNA. We have old DNA and new DNA as well. And but only in case of semi-conservative replication, you will find two bands. One is at the light position, N14 N14 band, and another is the N14 N15 band. So now let's learn about replication a bit in a bit more detail. So in replication, what happens? So replication means, just now we saw that the so just now we saw that the mode of replication in organisms is the semi-conservative mode of replication. So in semi-conservative mode of replication, what is happening? These the two strands of DNA separate, and these strands which separate are called the template strand. Okay, why are they called template strand? Because this from using this as the template or using this strand as the parental strand, the complementary nucleotides or complementary or a new DNA sequence will be produced, which is complementary to the original or the parental DNA strand, or whose sequence is complementary to the sequence of the parental strand. So how does this take place? So you have to understand. So what happens? New DNA is synthesized from deoxyribonucleoside triphosphate. So for replication, what are the things that will be required? So for replication, first you will require DNTPs, right? Dinucleotide triphosphates. That is the first thing that will be required. And then what are the other things that will be required? An enzyme is required, which is called as DNA polymerase. Okay. DNA polymerase is required and DNTPs are required mainly. Okay. Then there are other enzymes also which are required for unwinding of the DNA. There will be an enzyme called helicase. Okay. Then there are other multiple enzymes which are present during replications. You will learn them in week 5. But the main substrates or the substrates for main substrates for DNA replication are the DNTPs. So this is a structure of the deoxyribonucleotide triphosphate. So this is a deoxyribose sugar, and to this deoxyribose sugar, there is a base which is attached at the carbon 1 position, and the CH2OH, that the 5' position, you can see there are 3 phosphate groups attached. So that is why this, since 3 phosphate groups are attached, this is called DNTP. So deoxyribose. So deoxyribose, and there is a nucleotide because there are phosphate, sugar, and base. So there is nucleotide and triphosphate because there are 3 phosphate groups attached. Now you have to understand that during replication can only be synthesized in the 5' to 3' direction. And why is it so? Because this 3' in this 3' direction, why can it only extend in the 3' direction? Because this 3' end, this 3' end as a free OH group. Okay, this free OH group, and so what happens in replication? The 3' OH group of the last nucleotide on the strand attacks the 5' phosphate group of the incoming DNTPs. Okay, so this is the 5' phosphate group, and this is the 3' OH group. So this 3' OH group will attack the 5' phosphate group. So this is the one of the chemical reactions which you must have learned in organic chemistry also. So here this 3' OH group will attack the 5' phosphate group, and on this reaction, what happens? A phosphodiester bond will form between the 2 nucleotides. So this is the one nucleotide, this is the other nucleotide, a phosphodiester. This bond, this is called the phosphodiester bond, is formed between the two nucleotides and the 2 phosphate groups. These 2 phosphate groups are released. Okay. So this figure is again from the textbook peers. So in DNA synthesis, what happens? So that is why DNA replication can only take place in 5' to 3' direction, because this 3' OH group is free, and this 3' OH group attacks the 5' phosphate group of the incoming DNTP, and by this reaction, this PP or this diphosphate PPI, this inorganic dinucleotide phosphate is released, and a bond is formed between this OH group and this phosphate group. So in DNA synthesis, nucleotides are added to the 3' OH group of the growing nucleotide strand. So this is the 3' OH group of the growing nucleotide. This is the growing nucleotide strand, and these nucleotides are added. This 3' OH group of the last nucleotide on the strand attacks the 5' phosphate group of the incoming dinucleotide triphosphate. So this is the 3' OH group of the last nucleotide, and this will attack the 5' phosphate group of the incoming DNTP. So 2 phosphate groups are cleaved from the incoming DNTP, and a phosphodiester bond is created between the 2 nucleotides, the phosphodiester bond. So, so now direction of coming to the direction of replication. So how are both the strands acting as a template for synthesis of new daughter strand? So what happens that during replication, there is an enzyme helicase which will unwind the DNA. Now, if the DNA is getting unwound in this direction, okay, in this direction, so there will be 2 strands that will remain free, or 2 strands will be freed up, and so one of the strands, one of the template strand will have 3' to 5' will have this 3 prime to 5 prime polarity. Okay, so the strand which has 3 prime to 5 prime polarity, in this strand, what will happen? A new DNA molecule can be synthesized in this direction, 5 prime to 3 prime direction. Okay, because just now we learned that replication always takes place from 5 prime to 3 prime direction. So using the 3 prime to 5 prime strand as a template, this 5 prime to 3 prime, the DNA can be replicated in the 5 prime to 3 prime direction. Okay, from the left to right. So this strand is called the this strand is called the leading strand. This is called the leading strand. So what is happening in this strand? The my DNA is getting opened up in this direction. In this direction, unwinding is taking place in this direction, and the new daughter strand that is getting synthesized, that is also taking place in the same direction. Okay, in the same direction, the new daughter strand is also getting synthesized. So this is called the leading strand. While if you look at the other strand has polarity 5 prime to 3 prime. Okay, and the DNA replication can synthesis of new DNA strand can always take place from 5 prime to 3 prime direction. So what happens is so here you will learn about that RNA primers get attached, and then this 3 prime OH group is extended. Now once this start extending, there will be this 5 prime to 3 prime direction, the new daughter DNA strand will start getting synthesized. Now, as this strand is getting a new daughter strand is getting synthesized, more amount of this yeah, of this polynucleotide strand will become free, because this helicase is moving in this direction. Unwinding is taking place in this direction, but the DNA synthesis is taking place in this opposite direction. So as this new strand is getting formed, simultaneously what is happening, more polynucleotide strand is getting free. So after some point, what happens? The DNA polymerase has to start making new strands. Okay, new strands. So these are called the Okazaki fragments. Okay, so let's understand it one by one. So the new nucleotides are joined one at a time to the 3 prime end of the newly synthesized strand. Okay, so this is the 3 prime strand of the newly synthesized strand. Okay, so in this in the 3 prime OH direction, or the 3 prime free OH group, the new nucleotides will get attached one by one. So first nucleotide, then second nucleotide, third nucleotide, one by one these nucleotides will get attached. Now the DNA polymerases, the enzymes that synthesize DNA, can add nucleotides only to the 3 prime end of the growing strand, not the 5 prime end. So the DNA polymerases can add nucleotide only to the 3 prime end, because this 3 prime end has this free OH group. Okay, so only this OH group can attack the 5 prime phosphate group. So that is why nucleotides can be only added to the 3 prime end. It cannot be added to the 5 prime end. And so new DNA strands always elongate in the same 5 prime to 3 prime direction. It will always, the new strand which is getting synthesized, that will always be synthesized in the 5 prime to 3 prime direction. Now, as the DNA unwinds during replication, the antiparallel nature of the two DNA strands, so we had seen that the two strands of the DNA, they are anti-parallel to each other. Because of this anti-parallel nature, one template is exposed in the 5 prime to 3 prime direction. So this strand, you see here, this upper strand is exposed in the 5 prime to 3 prime direction, right? While this lower strand is exposed in the 3 prime to 5 prime direction. So the strand which is exposed in the 5 prime to 3 prime direction, so this strand is called the lagging strand, because here what will happen, there will be short fragments of DNA that will be synthesized. Okay, and these fragments are called the Okazaki fragments. While this strand, which has this polarity 3 prime to 5 prime, this is called as the leading strand. So in this strand, the DNA replication, or the new DNA strand can be synthesized continuously. While in this strand, the DNA synthesis will take place in a discontinuous fashion. This is also known as discontinuous replication. Here, what will happen? The replication will take place discontinuously. So because once this strand is a new strand is getting formed, there will be no more free DNA. So the DNA polymerase again has to move to that part of the DNA which has become free now. So it will move to the to that region, and it will synthesize a new DNA strand. So here there will be short short fragments of DNA that will be produced. So here in one strand, the replication is taking place discontinuously, while in the other strand, the replication takes place continuously. Continuous replication. Okay. So remember that the strand which is 3' to 5' polarity, and that in that template strand, the synthesis, the replication will take place continuously, while the strand which is polarity 5' to 3', in that strand, the uh, synthesis or the DNA replication takes place discontinuously. So this is also known as a lagging strand. The DNA polymerase synthesizes. So all this discussion was for you to answer this question. So the main thing that you have through the take home message from all this discussion is that the DNA polymerase synthesizes. So it is a DNA. So you have to answer this question. There are four options: DNA in the 5' to 3', 3' to 5', mRNA. So DNA polymerase, what it does, DNA polymerase. So it is a DNA polymerase means it is involved in DNA replication. What does DNA replication means? DNA polymerase, DNA to DNA, right? Replication. So these options are incorrect. So first of all, you eliminate C and D, because DNA polymerase means it is involved in replication, means DNA from one DNA, a new DNA molecule will be produced. Two DNA molecules will be produced. Okay. So the DNA. So just now we saw that the correct answer for this question is option number A, that DNA replication always takes place in the 5' to 3' direction, because the DNA polymerase can only catalyze reaction at the 3' or at the 3' end of the growing strand. Okay. Okay, so this is again through this figure, I've tried to explain that how these Okazaki fragments are getting produced, in how replication in one of the strand is discontinuous, while in the other strand it takes place continuously. So just look at this schematic. So what happens? So this is the lower strand. So if you study this figure very carefully, you will be you can easily understand what is happening in replication. So on the lower template strand, the DNA synthesis proceeds continuously in the 5' to 3' direction. So it takes place continuously in the 5' to 3' direction. On the upper template strand, the DNA synthesis begins at the fork and proceeds in the direction opposite that of the unwinding. So my unwinding is taking place in this direction, while the DNA synthesis is taking place in the opposite direction, in the direction opposite to that of the unwinding. So it soon runs out of template. Okay, so because my DNA is getting freed up in this direction, it is getting free in this direction, while this DNA synthesis is taking place in the opposite direction. So it is obvious, right, that after some point, the template DNA, the free template DNA there will be no free template DNA for the synthesis of new DNA. So what happens that the DNA synthesis starts again on the upper strand at the fork. So at the fork, again a new DNA synthesis. So there are very beautiful videos. You can go through them in YouTube. So how DNA replication is taking place. So you can easily understand by watching those videos that why one of the strands, the DNA replication takes place in a discontinuous fashion, and in the other, the DNA take place continuously. Okay. So DNA synthesis starts again on the upper strand at the fork. It will start at the fork and then it will again extend, each time proceeding away from the fork. So it is proceeding away from the fork. So DNA synthesis on this strand is discontinuous. So in the upper strand, the DNA synthesis is discontinuous. And here what happens, there are short fragments of DNA produced by discontinuous synthesis. So by discontinuous synthesis, there are short fragments of DNA getting produced. And these short DNA fragments are called the Okazaki fragments. So these are called the lagging strands. And this strand where continuous replication is taking place is called the leading strand. So you can read this. The same here thing is explained over here. So as the DNA unwinds, template that is exposed in the 3' to 5' direction allows the new strand to be synthesized continuously in the 5' to 3' direction. This new strand, which undergoes continuously, which undergoes new strand, which undergoes continuous replication, is called the leading strand. So this is called the leading strand. and the other template strand is exposed in the 5 prime to 3 prime direction. So you see here the template strand is exposed in the 5 prime to 3 prime direction. So after a short length of the DNA has been unwound, synthesis must proceed 5 prime to 3 prime, that is in the direction opposite to that of the unwinding. At each replication for synthesis of leading strand proceeds continuously and that of the lagging strand proceeds discontinuously. So the new DNA strand which is getting produced by using the 5 prime to 3 prime template, that is the called the lagging strand, right? The short lengths of DNA produced by discontinuous replication of the lagging strand are called the Okazaki fragment. Okay, so so why did I explain all this? So that you can understand this question. So what will be the correct option for this question? During replication, Okazaki fragments elongate, leading strand towards the replication fork. So you have to understand the concept very well for answering this type of question. So these are very confusing questions. And if you do not understand the concept, you can easily make a mistake. So the lagging strand away from the replication fork, leading strand away from the replication from lagging strand towards the replication fork. So the Okazaki fragments were forming in which in case of which strand? So the strand which had a polarity, template strand which had a polarity 5 prime to 3 prime. Okay. A strand which had a polarity 5 prime to 3 prime. So in that case, as the DNA polymerase can only elongate in the 5 prime to 3 prime direction. So the Okazaki fragments elongate the lagging strand. So the Okazaki fragments were present in the lagging strand and they were getting synthesized away from the replication spot. Okay. So if you go through the figure and if you understand the concept that I explained just now, you will not be able to answer this question. So in this direction. So these are the Okazaki fragments. They are lagging strand away from. So this is your replication fork and they are getting synthesized away from the replication fork. Okay. Should be correct. Now there is another last part of this today's lecture or this tutorial is Lamb-Brush chromosome. So this is one of the giant chromosome, and these Lamb-Brush chromosomes they are found mainly in the diplotin stage of prophase 1 of meiosis of the first meiotic phase. And these Lamb-Brush chromosomes they are present in the oocytes. They are present in the oocytes of vertebrates. In the oocytes of vertebrates, these oocytes remain in the diplotin stage for many years until puberty is reached. So in this stage, we can find that there are the chromosome that is present is has Lamb-Brush chromosomes. So these Lamb-Brush chromosomes they are transcriptionally active and they are present in large oocytes of vertebrates and invertebrates. And where does it take place? It occurs in the diplotin stage of prophase 1 of the first meiotic division in primary oocytes of all animals. So if you have a little bit idea how the oocytes are matured from primary oocyte to secondary oocyte to ovum, how they get matured, then you will understand that these oocytes get arrested in the diplotin stage of prophase 1. And once puberty is reached, then uh, these oocytes can proceed to further stages of meiosis 2. Okay, so they are arrested in this stage for around 10 to 12 years. So it is organized. So here, if you look at the chromatin in this stage, these chromatin are organized in a series of large chromatin loops emanating from a linear chromosomal axis. So this is one of the figure I took from the book Albert. Cell biology. So you can see here that there are loops which are extending from this long axis of chromosomal axis, and these chromosomes produce large amounts of RNA for the oocyte, and most of the genes are actively expressed. So from these loops, RNA can be transcribed. Okay, the DNA in loops remain highly condensed on the chromosome axis, where genes are generally not expressed. So the loop So these are the highly condensed chromatin. So then from this highly condensed chromatin, there is no RNA that is transcribed. While this extended chromatin in the loop domain, they are regions of active transcription. So this is what I was explaining. The Lamb-Brush chromosome, they occur in the diplotin stage of the prophase. So you remember this. So it occurs in the prophase 1, meiosis 1, or the prophase 1 of meiosis. It has prophase 1 of meiosis 1. It has 4 different stages. That is the leptotin, zygotin, pachytin, diplotin. Leptotin, zygotin, pachytin, and diplotin. So diplotin is the last stage of prophase 1 of meiosis of first meiotic division. So this Lamb-Brush chromosomes, they occur in this diplotin stage of prophase 1 of first meiotic division. So option number diplotin prophase 1 meiosis 1. So option number C is correct. Now this is one of the questions which we can take up uh, when you go through the lectures which are related to molecular biology techniques, mainly PCR. So if you go through the PCR reaction and understand the concept, at that after that lecture, we can take up this question. So once this PCR molecular technique is discussed, I will explain this concept. Basically, in PCR, you will find that there are 3 steps: denaturation, annealing, and extension. And this is one of the molecular techniques where you can amplify your DNA or gene of interest. So here, what happens is, so this question we will add because it will be difficult for you to understand now. We have not covered PCR yet. So I will take up the question in the later lectures. Okay. The answer is actually A, but we will discuss it in more detail once we cover this. This is covered in your lecture. This PCR technique is covered. Okay. And now, what are the amino acids where phosphorylation occurs? So in one of the lectures in the uh, third lecture of week 4, the several modifications which are found in histone proteins were discussed. Okay. Histone. So the histone tails are the what are the so the histone proteins they have the N-terminal regions. These N-terminal regions are disordered regions. They do not have a specific structures, and these N-terminal regions they remain free, and they are very they are very active sites for post-translational modifications. Okay, so this lysine, arginine, okay, lysine, arginine residues, they are very prone for post-translational modification. So these N-terminal regions of the histone proteins, these are called the histone tails. So the N-terminal tails of the histone proteins are called the histone tails, and they are very prone for post-translational modification. There are 3 main post-translational modifications which take place in the histone tails, which are the methylation, acetylation, and phosphorylation. So these are the 3 major modification which can be seen on the amino acid residues which are present on the histone tails. These things were discussed. So here in this question, what has been asked is which are the amino acids where phosphorylation occurs? Phosphorylation is basically when a phosphate group is attached to the any amino acid residue. So basically, so you have to remember that phosphorylation will mainly take place on amino. So where will phosphorylation take place? Phosphorylation takes place on amino acid residues, which have hydroxyl group as a side chain. So amino acids which are having hydroxyl group as a side chain. So there are 3 amino acids. One is the serine, threonine, and tyrosine. So if you see the structure of these 3 amino acids, serine, threonine, tyrosine, they have free OH group in their amino acid side chain. In their side chain, so the phosphorylation takes place in serine, threonine, and tyrosine residues. So all these 3 amino acid residues have free OH group in their side chains. So this is again from one of the lecture slides. These are directly discussed in your lectures. So the phosphorylation takes place mainly at serine, threonine, tyrosine residues. And who does this phosphorylation? There is an enzyme which is known as kinase. We have seen kinase when we were discussing glycolysis. So what do these kinases enzyme do? They transfer a phosphate group from ATP to the amino acid residue. Okay. Okay, so there are 3 main kinases: serine kinase, threonine kinase, and tyrosine kinase. So what will serine kinase do? It will transfer the phosphate group from ATP onto the OH group of the serine. Okay. So you see this figure. So this is serine kinase. Threonine kinase will transfer the phosphate group from ATP to the OH group of pre-OH group of the threonine. So this is threonine kinase, and tyrosine kinase transfers the phosphate group from the gamma phosphate of this ATP to the pre-OH group of tyrosine. Okay, so kinase transfer phosphate group from ATP to the hydroxyl group of the target amino acid residue. So if this is the target amino acid residue R, the hydroxyl residue or the amino acid residues which are having the free hydroxyl group in their side chain. So it adds significant negative charge that influences chromatin structure. So what will happen is once this phosphate groups are added, because these phosphate groups are negatively charged, it will repel the DNA, or the DNA from these regions will be open, because this negative charge on the phosphate group and the negative charge which is present on the DNA. Due to a negative charge which is present on the DNA backbone, they will repel each other, and the chromatin will get relaxed, or the chromatin will get decondensed if phosphate groups are attached. Okay, in more opening of the chromatin structure, leading to expression of more number of genes. So if a particular gene has to be expressed more, it is necessary that the chromatin gets loosened or gets relaxed in this region. So these type of modifications allow that portion of the of the DNA to get transcribed into mRNA and translated into protein. Okay, so that is all for today. Okay, so I hope now it will be easier for you to solve the assignment, and we will take up more questions in the next class. So if you have any doubts, you can we can solve those doubts, or we can take up those sections specifically. So thank you and have a good day.